Hello, everyone. It is my great pleasure to invite you all to this last webinar. As you know, we've been celebrating our 15th anniversary of Nature Nano for quite some time now. We started back in October, and finally, that's our last online event today. And we have two more exciting speakers that we invited today. And I just wanted to remind you before I introduce our first presenter that please ask questions. We have this questions box. You can also ask questions after each talk has been finished. You can ask directly our speakers, but if you want, just submit your questions and we will pass them on to our speakers. So our first speaker today is Professor Pablo Jarillo, who's from MIT, as you know, and whose experimental work on magic angle bilayer graphene had created quite a scientific revolution back in 2018 in condensed matter physics and in the field of 2D materials, of course, as well. Since the introduction of what is now we know as twistronics, these moiré systems formed of 2D atomic layers have been studied extensively, revealing their widely tunable electrical and optical properties and some exotic, strongly correlated and topological phenomena that they host, including superconductivity, correlated insulator states, orbital magnetism, to name just a few. So with that, I will pass you over to Pablo, who will bring you up to date on the recent developments of this very exciting field.
Okay, hello everyone. It's a pleasure to be giving this talk for the 15th anniversary of Nature Nanotechnology. I want to start by thanking Fabio and Chiara for the invitation, and Olga for the invitation and for the introduction. So today I want to tell you about the magic of moiré quantum matter. Okay, this is a field which has emerged over the past few years as one of the most vibrant fields of condensed matter physics and of nanoscience and technology. Okay, so let me start, however, from a more generic point of view. So among the most fascinating states of matter that we have in the universe are those where the interactions between the individual constituents that make up this matter are very strong. Okay, we call these strongly correlated states of matter. And this is a state of matter that occurs, for example, in the quark gluon plasma, where you have, which occurs just a few hundred microseconds after the Big Bang, and that we can recreate nowadays in collisions of heavy ions, for example, such as this one displayed at Brookhaven National Laboratory. The different phases of nuclear matter in neutron stars are also strongly correlated. Okay, this is a picture by the Chandra X-ray Observatory of a neutron star here at the center. And if you go to Wikipedia, you can see that these different phases, of nuclear matter in neutron stars, which are called, nuclear plasta, they are all strongly correlated phases. And if we go to, perhaps a bit closer to my field, to topological states of matter, you have, for example, states with very interesting topological properties, such as when you apply a magnetic field perpendicular to a two-dimensional electron gas. That system of electrons can go into the fractional quantum Hall regime, where you have very exotic quasi-particles with fractionalized charges. Now, when we go to strongly correlated quantum materials, these are materials where now the electrons are the ones that interact very strongly and can give rise to very unusual behavior. Perhaps the most famous family of strongly correlated quantum materials are the high temperature Cuprate superconductors. Okay, where in a phase diagram of temperature versus doping, there are all these unusual phases, such as strange metals, pseudo-gap phase, D-wave superconductivity, and even to this day, we have a relatively poor understanding of many of these phases. Now, when it comes to investigating strongly interacting systems, these are very hard to solve theoretically. Okay, so for example, let's take the cuprates as an example. In the cuprates, the key component is this copper oxygen plane, okay, where if you place 1 electron per copenatom or 1 electron per unit cell, you have this correlated insulator called a MOT insulator. Okay. Now, if you remove a few of these electrons, then the system becomes a superconductor. Okay, in this correlated insulator state, the electrons repel each other very strongly. They interact and repel each other very strongly, so they are all stuck in their positions. But now, as we remove a few of these electrons, the electrons can jump from side to side, but they do that in a correlated fashion that we believe gives rise to superconductivity. Okay. Now, even in the simplest model that we believe that captures the essential physics of the high temperature Cuprate superconductivity, okay, this is a model called the Hubbard model, where you take into account the repulsion that you for 2 electrons to occupy a single site, and you also take into account that the electrons can hop from side to side, via this tunneling T. Okay. Now, this model, as I said, we believe it describes the essence of the physics of high temperature superconductors. But I say we believe it because we do not know actually how to solve theoretically this model, and we don't know how to go from here to this phase diagram. Okay. Now, the difficulty in, for example, solving the system, strong interacting systems theoretically has led to the development of alternative platforms to investigate strongly correlated materials and also topological materials. Okay, so on one hand we have the actual quantum materials with lattice scales of a few angstrom. These are atomic scale materials such as cuprates, etc. Over the past couple of decades, we've developed, the atomic physics community has developed ultra cold atoms in optical lattices, where now the typical length scale is of order of a micron. You can shine lasers at each other. You can load them with atoms, and then you can tune the interaction strength between these atoms so that you can investigate strongly correlated physics and try to simulate a Hubbard model. And what I want to tell you about today is that, of the past few years, a new paradigm has emerged. It's called moiré quantum matter, with a typical length scale, we call the moiré length, of the order of 10 nanometers. So it's conveniently a couple of orders of magnitude in between, a couple of orders of magnitude from either of these other length scales. Yeah? And the energy scales are also complementary, intermediate between quantum materials and cold atoms. And we have also a degree of tunability and control over the systems, okay, which is also intermediate between these two. In cold atoms, the community has exquisite and extreme control over the system. In quantum materials, we don't have too much control. In moiré quantum matter, we have an intermediate degree of control. Now, excuse me. So initially, the technique and the breakthrough that enabled the development of this moiré quantum matter platform was what some people call twistronics, which is the fact that we can stack two-dimensional crystalline lattices on top of each other with an arbitrary angle of rotation or arbitrary twist angle. Okay, this is something that can dramatically modify the electronic, optical, and mechanical properties of the heterostructure that we make. Okay, there's no precedent in the history of material science about being able to do this at will. Okay, choosing any angle that we want, and it is only with the advent of 2D materials that we were able to do this. Okay, now, why would you want to study this platform, moiré quantum matter? Okay, so let me tell you a few reasons. One is that it is a very easy way to obtain isolated flat bands. And now these isolated flat bands allow you to investigate the interplay between kinetic energy interactions and topology in a way we didn't, we weren't able to do before. Now, as I will show you with a few simple building blocks, for example, graphene, or hexagonal nitride, or transition metal dichalcogenides, we can obtain a plethora of correlated and topological behaviors, okay? Which brings up the question of what are the key essential ingredients needed for complex emerging behaviors of quantum matter. Even that, with very simple chemical elements here, we can realize all of it. Now, it is an extremely tunable in situ platform. We can tune the parameters of the system and the phases of the system with electric fields, electrostatic doping, strain, pressure, magnetic field, temperature, and also tunable through the twist angle and the type of moiré system. And finally, this is a novel experimental platform to explore vast new families of hybrid materials based on non-equilibrium growth and assembly. Okay. So now, as I'm, those of you that have followed this, field will know, the magic a little bit of this moiré quantum matter is that we can realize almost pretty much all, behaviors of condensed matter physics, in this very simple platform. Okay, we can have correlated insulators, superconductivity, various topological phases, magnetism, pneumaticity, moiré ferroelectricity, and we keep discovering more and more phases of matter in the system. Yeah. Now, with this introduction, let me tell you what I want to talk about today. So I want to start by telling you about graphene and magic angle graphene, okay, which was the first moiré quantum matter system investigated. Then this gave rise to, moiré quantum matter, as I mentioned, and I will tell you a little bit about what were the developments that have happened since our discovery a few years ago, magic angle graphene. Then I'll tell you as an example of something beyond graphene, moiré magic beyond graphene. I'll tell you about moiré ferroelectricity in bilayer hexagonal boron nitride, and then I hope to have a few minutes to tell you about, just briefly, some outlook, some of the things that are happening in neighboring fields. So let me start with graphene and magic angle graphene. Now, graphene is a honeycomb of carbon atoms. Many of you are probably familiar with it. All of these atoms are carbon, so from the chemical point of view are identical. However, from the crystallographic point of view, we have two types of atoms, the A and B type of atoms, okay, or A and B sublattices, red and green atoms here. Now, one can calculate the electronic structure for electrons in graphene, the energy momentum dispersion, energy versus momentum in the X and Y direction. And as you can see, this is a very unusual electronic structure, okay? In particular, if you zoom in near the Fermi energy, you can see that the relationship between energy and momentum is linear, okay? So graphene has this linear energy momentum dispersion that has led to all these comparisons with massless ultra-relativistic particles. In fact, if you look at the Hamiltonian that governs this behavior, it's nothing else but the Dirac equation in 2 dimensions for massless particles. You can see here, energy is linear in momentum. In the usual Dirac equation, this is a spinor that tells you whether the electron has spin up or spin down. In the case of electrons in graphene, this is a pseudo spin, which tells you whether the weight of the electron wave function is on the A type of atom or on the B type of atom. Okay. Now, the other thing that I want you to know is that we have 2 of these, double Dirac cones. We call them valleys. They are the K and K prime valleys. So electrons in graphene have this 4-fold degeneracy, spin up and spin down, valley K and valley K prime. Just remember 4. It may appear later on. Now, when we put graphene on top of graphene, okay, we create a moiré heterostructure where the moiré wavelength, the distance between the soccer balls that you see here in the screen, depends on the twist angle. Okay, and in particular for low twist angles, is inversely proportional to the twist angle. Okay, now this is what happens when you put graphene in real space. What happens when you look at the momentum space or reciprocal space, and therefore what happens to the electronic structure? Yeah. So this is the situation already for a small twist angle between the 2 graphene sheets. Okay, this is the graphene Dirac cone of graphene sheet number 1. This is the graphene Dirac cone of graphene layer number 2. Okay, and as you can see, this is a situation that would be the case if the 2 graphene sheets did not know that each other exists. Okay, however, because when we put graphene on top of graphene, these 2 graphene sheets are just spaced by 3 angstroms. Electrons are very much aware, electrons in one graphene sheet are very much aware that the other graphene sheet exists. In particular, they can tunnel between the 2 layers. And that leads to this bonding, anti-bonding type of states, at this crossing energy, okay? This is very similar to bonding, anti-bonding states in a hydrogen molecule, but for a gigantic graphene molecule, okay? And this interlayer tunneling, leads to this band repulsion, in particular to this band that gets closer to, lowers its energy. This is the situation when the interlayer tunneling is much smaller than the energy of that crossing point, okay, which is given by this formula. Now, as you decrease the twist angle, this lower band here gets pushed down further and further low in energy until you reach 0, okay? When you reach 0, this is something known as a flat band condition. And this flat band condition is reached at the magic angle, which happens to be 1.1 degrees for graphene on top of graphene. Okay, so there was interesting, theoretical and experimental work about a decade ago about the single particle physics, essentially, of how this process happens and this magic angle, which was coined by Bistutzer and McDonald. Now, this is, before I showed you a cartoon, this is actually a real, calculation of the electronic structure for magic angle twisted bilayer graphene. As you can see, there are these flat bands. Okay, these are other remote bands, and these flat bands, they're not perfectly flat, but they're much flatter than the original graphene dispersion. Now, flat bands in momentum space means that these are, if you look at the electrons, where are the electrons in real space that are in these flat bands? They are highly localized in real space. To go from momentum space to real space, you just have to do a Fourier transform. Okay, so you can see that if you look in real space, where are the electrons when they are situated in these flat bands? They are in regions of AA stacking. Okay, where locally all of the carbon atoms in layer 1 are on top of all of the carbon atoms in layer 2, and this is separated by regions of, due to the small twist angle, regions of AB and DA stacking, where the electrons do not like to be. So from a schematic point of view, magic angle twisted bilayer graphene is a system where you have these AA regions where electrons like to be, and they are separated by AB and DA regions where the electrons do not like to be. In a slightly more realistic schematic, these AA regions, which are denoted here in yellow, they're separated by about 13 nanometers, okay? And electrons like to sit here, so this is going to form the equivalent of a triangular Fermi-Hubbard lattice in cold atoms, except that it's not really triangular, it's a honeycomb lattice because these A, B, and BA regions are not identical. And it's also not, this magic angle bilayer graphene has a special topological, structure that does not make it really analogous to a Fermi-Hubbard lattice, but you can think of it a little bit like that. Now, what my group discovered back in 2018 is that if you make magic angle twisted bilayer graphene devices and you position your Fermi level, your chemical potential in these flat bands, you can find an unusual set of correlated insulator states, insulator states which are due to strong electron-electron interactions. And then if you dope a little bit away, with charge carriers from that point, you have actually electrically tunable superconductivity. Okay, so magic angle twisted bilayer graphene superconducts. Okay, this was the first time that purely carbon system, okay, which was just electrostatically doped, was made. A purely graphitic system became superconductor. Now, this occurs only for a narrow range of angles around 1.1 degrees. Okay, and if you move further away from that region, then you don't really see this behavior. Now, magic angle twisted bilayer graphene is an electrically tunable superconductor. Okay, that means that when we put a certain number of electrons per unit cell in our system, we get a correlated insulated state. And now, as we add electrons or holes or electrons, which we can do electrostatically in our system, because we form a parallel plate capacitor between the magic angle graphene and a metallic gate. So we have a field effect transistor geometry, and we can change the number of carriers electronically. Okay, we can realize this electrically tunable superconducting nodes. Now, when my students showed me this data, immediately it reminded me of the hundreds of times I have seen phase diagrams such as this. Okay, this is a phase diagram of the Cuprate superconductors. At zero doping, you have a mode insulator state. That actually means 1 electron per unit cell. Okay, in our case, it happens to be that we have a correlated insulator state at 2 electrons or 2 holes per more unit cell. And now let me flip this axis, okay, so that they are identical to here. As you can see, if they hold dope, they have a large superconducting dome. If they do electron doping, they have a smaller superconducting dome, very similar to this behavior, okay? A big difference, of course, is that in order to realize this phase diagram, this is a theoretical phase diagram. In order to realize it, you need to fabricate hundreds and, grow hundreds and hundreds of crystals, each for each doping on this axis, and cool down many times and measure all of this data, okay? In our case, however, we measure this continuously because we can vary the doping electronically by dialing and knobing a gate, okay? And we can go from here to here just in a few seconds. Now, these experiments gave rise to sort of the field of moiré quantum matter. We posted this in March 2018. The paper was officially published in April 2018. And then what I call the theory tsunami came, okay? So there was immediately hundreds and hundreds of papers. This is just some of the papers that appeared in the first few weeks after we published our results. And people essentially were very interested in trying to understand what's the origin of the correlated insulator states and what is the superconducting order parameter, okay? All kinds of proposals with all letters of the alphabet, as you can see here, were proposed: S, D, S plus P, S plus P plus ID, etc. Okay, we do not know. The answer is we do not know yet, although evidence is built in that the order parameter may be quite unconventional. The superconductivity may be quite unconventional. Okay, now, at the same time, the popular press also took a liking for this field. So lots of papers and articles appeared in magazines of various types, about the system. So this was quite an interesting development. Now, let me tell you more interestingly about what happened since we published our results. Okay, so first thing is that we have reproduced our results. We have made a lot more devices. That doesn't always happen, so it's good when it happens. Okay. So now we have many, many more devices, and we're starting to obtain this, superconducting critical temperature as a function of twist angle. This is a new parameter, a new knob in condensed matter physics, the twist angle, and we can calculate properties as a function of this knob, twist angle. Now, even more important than, we're reproducing our results is when other groups independently reproduce our results, only then it becomes science, and this has happened thankfully by many, many groups. Okay, the first one was the group collaboration between Andrea Young and Corey Dean, which not only reproduced our results, but extended them in very interesting directions under pressure, and then the group of Dmitry Efetov later on. By now, robust superconductivity has been reproduced and extended by about a dozen groups, and more groups keep joining. Okay. Now, plenty other phenomenology characteristic of quantum materials has been observed. For example, strange metal behavior or pneumaticity. We have discovered other correlated systems in this moiré platform, for example, ABC aligned to hexagonal boronitride or magic angle twisted bilayer graphene. Okay, lots of local probe studies, so scanning probe microscopy studies have appeared. These were very interesting because people could start to look microscopically at magic angle graphene related systems. In fact, back in 2019, there were these 3 papers appearing in Nature simultaneously, also another one in Nature Physics. Let me give proper credit to this one too. Okay, and they started to look at microscopically the system, and they saw, for example, they were able to see that there is an increased bandwidth of the system when the Fermi energy is in the flat bands. Okay, that means that the bandwidth is set by the interactions. There was also evidence for pneumaticity, and people have been displaying more and more, looking at more and more interesting behaviors. Recently, there was even a spectroscopy of the superconducting gravity systems. Now, another very interesting development was that the community, has observed, ferromagnetism, anomalous Hall, and even quantum anomalous Hall physics in the systems, which means that, topology constitutes a centerpiece of this whole field of moiré quantum matter. Okay, in fact, this field of moiré quantum matter has meant the merging of several condensed matter communities. On one hand, the 2D and advanced materials community and heterostructures community, like people like me, which were working on graphene. On the other hand, the strongly correlated materials community, people that were working on cuprates, pnictides, etc. And then the people that work on topological condensed matter physics, people working on quantum Hall, fractional quantum Hall, topological insulators, valence semimetals. All of these communities have come together in this field of moiré quantum matter. Okay? Back in 2019, together with my collaborators at MIT, we already predicted theoretically that there would be nearly flat term bands in various types of moiré superlattices. Okay? Then soon after, the group of David Haber-Gordon at Stanford reported experimental discovery of very large anomalous Hall effect in magic angle twisted bilayer graphene aligned to hexagonal boronitride. And then about 6 months later, the group of Andrea Young reported actual quantization, the quantum anomalous Hall effect in this system. Okay, so this was, of course, a very interesting development. There was quite a bit of other theoretical work, as well as work on ABC aligned to HBM. Now, we have realized, pretty novel types of quantum devices. For example, hypertunable Josephson junctions. So this is a paper actually that we published this year in Nature Nanotechnology that I was very happy with. So in using magic and graphene, since we can control electronically the phase diagram, we can make for the first time the following type of Josephson junction. Now, Josephson junction is a superconductor insulator superconductor type of junction or superconductor normal metal superconductor type of junction. Usually in a Josephson junction, the superconductor is a different material than the insulator or the normal metal, okay? And they are typically fixed. There have been a few experiments where people have been able to tune the middle piece, okay? The cheese in the sandwich, so to speak, okay? For example, you can make graphene Josephson junctions where the superconductor is fixed, and the graphene you can tune electrically. In magic angle graphene, because we can tune electronically the magic angle graphene between superconductor, normal metal, and correlated insulator phases, it turns out that by playing with local gates, by putting different metallic electrodes to control the density locally, laterally, we can realize SIS or SNS junctions, okay, Josephson junction, where both the superconductor and the cheese, the normal piece or the insulator, can be controlled electronically, okay? This is the first time that such an electrically hypertunable Josephson junction has been made, and we can make various types of, Josephson junctions and look at Fraunhofer patterns and the different characteristics, study the two-dimensional, the non-local Josephson effect, which occurs in 2 dimensions, and we can even tune the system differently to make, for example, single electron transistors as it is shown here, okay? So this is something that gives extra versatility to this type of Josephson junctions, which are, it's unparalleled, there's no other counterpart using any other type of superconductivity or superconductor. Now, the next thing that I want to tell you is that we have, realized the next generation of moiré quantum matter, okay? Based not on bilayers, but on multi-layers. I call this moiré magic 3.0. So earlier, this year, the group of Philip Kim and my group too, we reported back-to-back in Science and Nature the discovery of a second moiré superconductor, okay? Is magic angle twisted tri-layer graphene now, 3 layers twisted on top of each other, okay? So it's quite interesting. It's an even more tunable superconductor than magic angle bilayer graphene, because now we can tune it also with a transverse electric field, and it realizes a strong coupling limit of superconductivity, okay? In fact, when physicists compare how strong are superconductors, typically one looks at what is the value of the critical temperature versus the Fermi temperature, which is like the density of electrons, okay? Now you can have different limits in 3D or in 2D, okay? And basically in this diagram, conventional superconductors tend to be near this corner. For example, aluminum has a 1 Kelvin of a TC of 1 Kelvin and a Fermi temperature of 100,000 Kelvin, okay? Given how many electrons aluminum has, it superconducts very modestly. As we go towards this diagonal, you find more and more unconventional type of superconductors. In fact, this purple band contains, the cuprates, the pnictides, organics, the heavy fermions, pretty much all the unconventional superconductors that we know. Now, magic angle twisted bilayer graphene and magic angle twisted tri-layer graphene, depending on whether you take the 50% normal state resistance or the Berezovsky-Costa-Litaules transition temperature, they correspond to these data points here, okay? So as you can see, they are among the strongest, actually the strongest coupled superconductors that exist, okay? Only recently another, superconductor, lithium zirconium nitrogen chloride, from Walsh's group, has demonstrated also this limit of ultra strong coupling. Now, another thing, an interesting development has been that we have realized that magic angle twisted tri-layer graphene is not a spin singlet superconductor, okay? Conventional superconductors are spin singlets, spin up, spin down, okay? Magic angle twisted tri-layer graphene, if we measure the superconducting dome, okay? Temperature versus doping. And now we look at what happens when we apply a parallel magnetic field so that we can look at the effects of Zeeman field on the system, okay? Usually a spin singlet superconductor would disappear at a certain, the superconductivity would disappear at a certain magnetic field called the Pauli limit. But it turns out that in magic angle twisted tri-layer graphene, the system superconducts way above the Pauli limit. These data points represent here the Pauli limit for different resistance thresholds. And this dark blue here, which means superconductivity, shows that the system is superconducting up to magnetic fields much, much larger, over a factor of 3 larger than the Pauli limit, okay? Not only that, but we can, in some parameter regimes, we can actually kill superconductivity, and then superconductivity reappears at high magnetic field. This is known as reentrant superconductivity, and it typically only occurs in very exotic type of ferromagnetic or spin triplet superconductors, okay? So magic angle twisted graphene is very likely not a spin signet superconductor. Now, let me just tell you in the next few minutes about an example of moiré magic beyond graphene, okay? And this is moiré ferroelectricity in bilayer boron nitride. So, as I mentioned earlier, we have all these behaviors of moiré quantum matter and all of these phases, and until recently there was one phase missing, ferroelectricity. But earlier this year, moiré ferroelectricity was reported by 3 different groups simultaneously, okay, among them my group, so the group of Moshe and Shalom, and also work at Manchester. So now, 2-dimensional ferroelectrics are extremely rare, okay? I think basically most of them, if not all of them, are actually in this slide, okay? And typically these are, okay, materials which have been obtained by exfoliating bulk polar materials, okay? Now, the thing that I want to talk about is very different. It's making ferroelectricity out of non-ferroelectric materials, okay? So the example that I want to think about is bulk bilayer hexagonal boron nitride and compare it with parallel stack bilayer boron nitride. So bulk boron nitride is a material which has a stacking which is known as A prime stacking. It is centrosymmetric, okay? You alternate the boron and nitrogen atoms in the crystal structure. It's similar to graphene, but now with boron and nitrogen atoms in the A and B sublattices, and they alternate in the vertical direction. So natural stacking has hexagonal boron nitride layer number 2 rotated by 180 degrees on top of layer number 1, okay? Now, if we take a single layer of boron nitride and now we break it, okay, let me point to the locations of the atoms, boron and nitrogen. Now we break it and we stack it parallel to itself, okay? So with 0-degree angle between layer 2 and layer 1, now you can see that nitrogen atoms are on top of nitrogen atoms, boron on top of boron. Now, this is a non-centrosymmetric arrangement. It's also very uncomfortable for the system, okay? So the system decides to shift towards AB stacking or BA stacking, okay? And it turns out that these 2 configurations, and AB and BA stacking, have electric dipole moments which are opposite of each other. So if we can switch somehow between these 2 configurations, we have a switchable ferroelectric, okay? And indeed, this is something that you can do, okay? So we have fabricated parallel stack bilayer hexagonal boron nitride. We place graphene on top because in order to investigate the ferroelectric state of the boron nitride, we're going to build a ferroelectric field effect transistor where the configuration of the parallel stack boron nitride, the electrodipole moment will lead to a change in the type of carriers in the graphene, and we will see that in the resistivity of graphene, okay? And indeed, if you make this type of devices, and now you measure the resistivity as a function of electric field, you see that you obtain hysteresis when you sweep up and when you sweep down as a function of electric field. And this hysteresis, okay, is indicative of this ferroelectric behavior, okay? Now, we can study this continuously as a function of bottom gate and top gate, okay, that we have in our structures. We can see the abrupt switching from BA to AB type of stacking. Then we can reverse and observe the opposite switching. Sometimes we can actually catch a domain wall boundary, okay, a domain wall as it goes through our contact electrodes. So for a little while, there is actually this double peak in the resistivity, indicating the presence of 2 domains. Yeah. Now, that is for perfect parallel stacking. If we have a small twist angle between the 2 layers of hexagonal boron nitride, now we have these BA and AB type of domains, similar to magic angle graphene, okay? Now, these domains have different energy configuration and different displacement, electric dipole moment. So when you apply an external electric field, if it is of one type, you will increase the size of one of the type of domains. If you have another type, you will decrease the size of the other domain, okay? And in fact, if we realize, twisted bilayer boron nitride, in this case 0.6 degrees, and we make a similar ferroelectric field of the transistor, we can see that at a given electric field, one type of domain configuration is dominant, and then slowly we switch towards the other type of domain configuration, okay? This is in contrast to when we have parallel stacking of the two boron nitride layers where the switch is abrupt, okay? You can see here the gradual transition from one domain type to the other domain type. Now, do we have direct, evidence imaging of this moiré ferroelectricity? Indeed, we can do vertical piezoelectric force microscopy to look at this moiré ferroelectricity, okay? And in fact, if you look with a vertical piezoelectric force microscopy, you can see both in the phase and in the amplitude images, this beautiful moiré pattern, okay? This beautiful honeycomb moiré patterns, which you can lay on top of here, and you can see this moiré ferroelectricity, okay? And I should mention that similar work was also obtained by the other two groups, and related work a little bit earlier at Columbia University. Now, this ferroelectricity is very robust. It survives at room temperature, okay? So this may actually provide opportunity to have real applications. I should mention that this thing can be done not only with hexagonal boron nitride, but with any bipartite lattice. And recently we have a paper now, which has been accepted in Nature Nanotechnology, with demonstrated this behavior in transition metal dichalcogenides. Okay, this paper is not yet published, but I want to show the paper. Now, in the last couple of minutes, just let me tell you a little bit about Outlook. Okay, so as I have told you, this moiré quantum matter is a little bit of a magical, field where you can realize, all kinds of phases, just using these very simple components such as graphene or hexagonal boron nitride or transition metal dichalcogenides. But we have hundreds and hundreds of two-dimensional van der Waals materials, okay? So we have barely started to scratch the surface of this moiré quantum matter universe, okay? The building blocks can be, for this moiré quantum matter, can be much more exotic. For example, superconductors themselves, such as ion diselenide or high-temperature cuprates, ferroelectrics, quantum spin liquids, etc. In fact, the community is starting to, the theories are starting to predict what kind of things you can get when you put together more exotic blocks, okay? So two examples of this are, a recent work by Marcel Franz predicting high-temperature topological superconductivity in twisted double-layer copper oxide. Okay, when you take two monolayers of high-temperature copper superconductors and you twist them, you can have topological superconductivity with an exotic on the parameter. And the group of Leon Valens has, published a series of papers on what happens when you twist two-dimensional crystalline magnets. Okay, they can form so-called moiré skirmions and all kinds of interesting novel spin textures. Now, there is also moiré magic beyond twistronics, or I should say beyond electronic systems. So for example, the cold atoms community is starting to calculate moiré and twisted cold atom lattices. What happens when you realize, this with cold atom systems. There is also something called moiré and twisted phononics, where you can realize, all, these single particle effects, these, huge enhancements of the density state using vibrating plates of phononic systems. And you can, of course, do this also using photonic systems. So you have moiré and twisted photonics, and people have realized all kinds of, interesting photonic lattices where you can, slow light very much, by having an appropriate twist angle, okay? So with this, I want to end my talk. I want to acknowledge my group members. This was largely the work of Joan Sao, Daniel Rodan, and Jane Park for the graphene systems, and Kenji Yasuda and Shiri Wang for the hexagonal boron nitride ferroelectricity, together with plenty of collaborators at various institutions. And I hope that we can soon go back to this type of events in person, and I would like to invite you all want to join me for a nice paella. I also want to thank our funding support, and of course, thank again Nature Nanotechnology. Congratulations on 50 years of Nature Nano. It's a great journal, and I hope you continue for a long time. I thank you. I want to thank you all for your attention. Thank you very much. I'm happy to take questions.
All right. So Pablo will join us now for questions. So please raise your hand if you want to ask a question. But before somebody, gets courage to do it, I will ask a question I wanted to ask. I was just thinking that the combinations are completely endless, and we can see it now. So the major problem is, I guess, to find this non-trivial kind of phenomena from what theory predicts, right? Because trial and error is not really an option for you. And so now we have bilayer graphene, we have trilayer graphene, superconductivity. Can you just tell us a little bit, or especially non-specialists, what are the main prerequisites or properties that the system must have to kind of exhibit, let's say, superconductivity properties, potentially, from the theoretical point of view?
Mm-hmm. So this is a question that is not clear. We do not have a clear answer to this day. There is a theory, a theoretical proposal, that is what inspired us to look, for example, at trilayer, which says that a symmetry of the system is important. It's a 180-degree rotational symmetry around the z-axis. So if you prepare a system with alternating layers with the exact same twist angle, these theories predicted what is the angle that you need in order to have flat bands, and presumably in these flat bands, when you put your chemical potential there, you will have superconductivity. So this theory works for 2 layers, it works for 3 layers. Now we have to see if it works for 4, 5, 6, 7 layers. But if it did work for those numbers of layers, it would perhaps give a little bit of more credibility to that theory. Perhaps indeed this symmetry is essential.
So that you're talking about graphene, right? And what about beyond graphene?
Well, superconductivity has
you mentioned like copper oxides, for example. So there are other systems obviously can exhibit.
Superconductivity has not been found in any other moiré systems, only in graphene-based moiré systems. So the community is still trying to see if a variety of correlated states and quantum anomalous Hall and so on has been seen in other moiré systems, but not superconductivity. So the community is still waiting to see a little bit if, for example, the fact that graphene systems have these spin and valley degrees of freedom, which are extra degeneracies, if that gives them more wiggle room to realize, an ultimate phase which is superconducting compared to transition meta dichalcogenides. The truth is that we don't know yet. A lot of people are exploring other moiré systems to see if we can find another superconductor which is not graphene based, but we haven't succeeded yet.
Okay, thanks. And I also wanted to ask you, because I'm not a condensed metaphysics scientist, so I'm always looking at applications. I wanted to ask you, I think I asked you already before, but not for our audience today. What are the applications that you can potentially see for twistronics, if any at all? Because maybe it's just a matter of fundamental studies, and that's where it's going to end in the end.
I think that for now, the community is very excited about just the fundamental knowledge that we're gaining. It's a new materials platform, it didn't exist before. We're having a lot of fun with it. Now, having said that, I mean, if I show, the data that I showed in the seminar today, at a very simplistic level from an engineering point of view, magic angle graphene is a superconducting field effect transistor, a transistor which I can turn between superconducting state and off state. So any application that you can think of where a superconducting field effect transistor might be good, then magic angle graphene is actually a pretty decent candidate to be a viable technology. That is, if we were able to fabricate these type of devices at scale, which I don't think it's impossible, but I think it would be hard. It will be challenging. But there are some groups already that are trying this. Now, that's on a more like, quantum computer, cryogenic, classical superconducting platform type of. Other thing that you can do, which there are already a few papers, is that these systems can be potentially good catalysts. There's a paper already posted about interesting electrochemistry, because you're modifying tremendously the density of states of these systems, and people are calculating, and they can be actually much better catalysts for example, for hydrogen extraction, out of water and so on, than the best existing catalyst that we have. For now, this is all theoretical work. We have to do the experiments, but that's another route that perhaps it might be interesting to explore. Again, initially, we will just do it on small devices and see if it works at all. Catalysis is a field that, you need, typically you speak about tons of catalysts. So it will be again, challenging to do this. But I always trust my engineer colleagues. They're very smart. If there's enough motivation, I think they'll figure things out.
Yeah, in this paper that probably I should be talking about, but you already mentioned it in your talk on TMDs, on the interfacial ferroelectricity and TMDs. I want to ask you how easy it is for people that, let's say, want to go the same path to control those tiny angles that you're using in your experiments, because they are like really precise. And is it really critical in order to observe the domains, for electric domains?
So that's a very good question. So indeed, for this interfacial ferroelectricity, whether it is with hexagonal nitride or with transition metal dichalcogenides, which gives you many more options actually, and it also works at room temperature, the twist angle is not essential. It can be zero, it can be a small twist angle. You will see ferroelectricity. It's not as delicate an effect as the magic angle graphene. So in that sense, one could envision, and this thing works at room temperature. So thanks for reminding me about that. One could envision, growing these semiconductor films, slapping them on top of each other at even zero angle. Then you get ferroelectricity, and you could, cut it, laser cut it into small domains and make hundreds of such devices, which perhaps, ferroelectrics are now very popular in the nanoelectronics community to help with subthreshold slopes in field effect transistors, but also for membristive neuronal networks, etc. So I think I have a few colleagues that asked me about this in the electric engineering department, like, oh, wow, you have a new type of ferroelectric. Let us play a little bit with it and see what we can do. And I think that might also be perhaps a nearer term type of application than superconducting devices or catalysis, for which fabrication is a more challenging process.
Yeah, exactly. We also have 2D magnets, that we can twist and turn. Okay, I think we're out of time. Thank you so much, Pablo, for being a very valuable author and reviewer, and for, just remembering about Nature Nano when you submit your great work. So thank you very much.
Thank you.
Thank you for joining, for doing it, and for celebrating with us. And it's kind of we are wrapping it up with you almost. So we still have Polina, of course. All right. Thank you. Thank you very much.
Thank you, everyone.
Thank you. Okay, then it's over to Chiara now, and our second speaker.
Yeah, hello, here we go. Hi, thank you, Olga, and thank you, Pablo, for this really nice talk. And we have our second speaker with us today, and we're really excited to have Paulina Anikeva. She is a professor of material sciences and engineering, as well as brain and cognitive science at MIT, where she leads the bioelectronics group. She has a PhD in physics, and she's actually started working with solar cells and then moved to work with light emitting devices with organic material and quantum dots. And then she moved during her postdoctoral studies she was interested more in biology, and so she moved towards that side, working with Carl Disseroth on neurobiology at Stanford. And there she worked on optoelectronics devices that could tune neural signals, and that's what she continues to work on in her lab today. So her interests are on developing magnetic materials and optoelectronics devices that can be interfaced with the neural system, and that can be used to tune or to record as well neural signals in the brain or in peripheral systems. And the idea is to try and understand better neural signaling, but also to provide some tools and technological tools to maybe offer some therapeutic avenues for diseases of the nervous system. Paulina, thank you very much for being with us today and for concluding actually our series on the 15-year anniversary of Nature Nanotechnology, and for concluding also our Nature Nano Talks for the year. It's really a great pleasure. I'm looking forward to hearing your talks, and I will see you afterwards for the questions. And so that's over to you.
Good afternoon or good morning, everyone. I'm going to share my screen. I believe I'm already sharing, so I have a webcam, and can you see my slides, Chiara? Thank you so much for the kind introduction, by the way. You can see my slides. Okay, perfect. So let's go into full screen. It is my absolute greatest pleasure and big honor to be concluding this anniversary series of talks for Nature Nanotechnology. It is a wonderful journal for our community, and I'm going to share some of the work that our group has been doing in using nanomaterials to modulate the neural signaling and understands neural circuits. So what brings someone like myself from a field of nanotechnology and optoelectronics to neuroscience? Well, it has to do with the absolutely unimaginable beauty and complexity of the nervous system. If you think about the sheer multitude of scales that we need to consider in the nervous system, it starts to get dizzying. First, if we take a look at circuits, let's say at the level of the brain or at the peripheral circuits, we're now talking about signaling at the scale of centimeters and millimeters. Or if you're talking about the periphery, you can actually think about kilometers of axon fibers going through our body. But if we want to start zooming in onto how the neural circuits actually work, we need to start considering individual cells. So neurons are the signaling molecules of the signaling cells of the nervous system, but there are other types of cells that are also important, and all of them operate at the micron scale. But now, ultimately, what makes us human, what encodes our physiology and our behavior, it's the signaling between the neurons and the wiring between the neurons. So let's zoom in another level, go to submicron scales, and we start seeing synapses. And at synapses, neurons can exchange neurochemicals and ion fluxes, which ultimately results in neural activity. But that's not it. We need to zoom in one more level, and that level is of proteins, ion channels, receptors, pumps that allow us to transport ions molecules across the membrane. And those receptors can respond to a variety of signals, and they can convert those signals into neural firing or neural silencing, ultimately controlling the activity of neurons, activity of networks, controlling our physiology, controlling our behavior, and making us who we are. So how do we make this link across all of those scales, and how can we leverage nanotechnology to make the connection between the function of receptors that operate at the nanoscale to function of whole organisms that, of course, function on the macro scale? How can we go from molecules to behavior and physiology? And our group is inspired by nanotechnology and thinks about how can we modulate biological signaling without actually perturbing the function of whole organisms. How can we get at individual receptors without perturbing anything else? And for that, we leverage magnetic fields, because magnetic field is the only modality that can penetrate through biological matter completely unattenuated and without deleterious effects. And the reason for that is that we are, of course, weakly conductive, and also magnetic susceptibility of biological matter is negligible. So weak magnetic fields that are on the order of millitesla or tens of millitesla go through us and can go into arbitrarily deep portions of the body. But now, how can we convert those magnetic fields into something that biology can actually understand? And here, nanotechnology comes up with solutions. For instance, we can synthesize magnetic nanotransducers with various properties that can convert magnetic fields into signals that can be perceived by receptors on neural membranes. For instance, we can start simple by converting magnetic fields into thermal stimuli by leveraging magnetic hysteresis in nanoparticles of biocompatible metal oxides. We can take this method one step further and leverage heat dissipation from magnetic nanoparticles to design a chemical delivery schemes that will allow us to modulate chemoreceptors. But alternatively, we can go away from heating and instead leverage movement, mechanical either displacement or torque that magnetic nanomaterials can apply to neural membranes in response to weak magnetic fields. So we're going to explore all of these modalities in the work that our group has conducted over the past decade. So let's start simple with magnetothermal transduction. Here we will be relying on hysteretic heating of magnetic nanoparticles in weak but rapidly alternating magnetic fields. This work has been started by a talented former student, Richie Chan, in our group. So when we're talking about hysteresis, if we are considering magnetic nanoparticles that are about some tens of nanometers in diameter, we'll pick it such that it is a single domain magnet and it will have a magnetic moment. This magnetic moment can follow the direction of magnetic field, but as we try to switch the direction of magnetic field, the magnetic moment has to overcome magnetocrystalline anisotropy, and as a result there is a lag between magnetization and magnetic field. That lag manifests in the hysteresis loop, and the area of the hysteresis loop is proportional to the heat dissipated by the particle. And if we change the frequency, we can have more cycles of this heat dissipation. So now we have an ability to produce heating, and that's a fairly standard method called magnetic hyperthermia that existed for decades. But rather than applying this method to, let's say, kill tumor cells, which has been done for about 50 years, instead we are going to tune our heat carefully such that we can trigger specific ion channels. And in fact, the ion channel that we will demonstrate this on is something that's quite familiar to all of you, especially when you're thinking about drinking hot tea or eating spicy food. Every time you eat hot chilies, you get a sensation of burning pain on your tongue. And the reason for that is the capsaicin receptor, TRPV1, that is expressed in your cells in your tongue, but also is expressed across your nervous system. So this same receptor responds to both capsaicin and heat. And if we sensitize neurons to this receptor, we can then surround them by magnetic nanoparticles and then apply magnetic field. And only if our cells are sensitive to heat will we see them firing in response to this really weak but fast magnetic field through magnetothermal stimulation. This is, of course, movies that we have recorded in a dish, but we can extend this application and show it in vivo, in live animals. In this case, applying this magnetic hysteretic modulation to a deep brain region called ventral tegmental area in mice. This region is hard to access because it's quite far into the depth of the brain, and it's also really small. There's no natural heat sensitivity, but we can enable that heat sensitivity through genetic modification. And then we can show that only when neurons are sensitive to heat and the nanoparticles are present in this brain region, only then will magnetic field trigger neural activity. In this case, magnetic field is applied to the whole animal. And this work has been recently extended in collaboration between our group and a wonderful group of Yasin Temel in Maastricht University, led by talented postdoc Sarah Hesham, who has applied this method to stimulate neurons as a potential therapy for Parkinson's disease. So what Sarah has done is that she has had Parkinsonian animal model that was achieved through oxidative stress of a group of neurons called dopamine neurons in deep brain region called medial forebrain bundle. And when those neurons die in people, that's typically manifests in Parkinson's, which is poor control of movement. And in this mice, we also see poor control of movement. They typically fall off an instrument called the rotarod. They also have very irregular gait, very irregular stepping. But if we inject magnetic nanoparticles into a region called subthalamic nucleus, which is the region where we traditional clinical electrical stimulation implants are inserted in human patients. If we replace those implants with our nanoparticles and apply magnetic field, we can restore the ability of these mice to move. They will be able to hold on to the rotarod, and they will have regular stepping just as normal mice, giving you a little bit of a therapeutic flavor that technology has in its potential. But we don't need to limit ourselves to direct stimulation of thermal receptors. We can extend this hysteretic heating to controlling other molecular processes. For instance, we can control phase transitions in lipid carriers. For that, we can design a liposome that is stable in physiological temperatures such as 37 degrees Celsius, but undergoes liquid crystal transition and becomes porous at 42. And that allows us to release water-soluble payloads from those liposomes. So here is a transmission electron micrograph of our magnetic nanoparticles introduced into the core of the liposome along with a small molecule. And we can demonstrate to ourselves that indeed application of the magnetic field allows us to release these payloads, here shown through a release of a dye, and that release can be controlled either through different time period of application of the field or through different magnetic field magnitude and other parameters. And this work was led by a talented postdoc, Xian Rao, who is now a professor in the University of Massachusetts. And then we can apply this method to, again, control neural activity. In this case, packaging a molecule called clozapine and oxide, which happens to be a ligand for a class of receptors called HM3DQ. In this case, we can see that every time if our liposomes are loaded with this molecule and we apply magnetic field, it will trigger these receptors, triggering calcium influx into neurons. Here, every line represents a neuron, and we have hundreds of them, and brighter scale means more calcium flowing into those neurons. But if our liposomes are empty, or we don't apply magnetic field, or we don't have expression of this receptor in our neurons, we do not see this type of neural excitation. We can alternatively apply this method not only in a dish to control these receptors, but also to control these receptors in the context of behavior. In this case, actually, choosing again a deep brain circuits that connect ventral tegmental area that we have briefly talked about, and the other area called nucleus accumbens. This circuit is responsible for reward as well as social preference in mice as well as humans. And if we stimulate this circuit, mice can become more social. And the way we test it, if we put mice into a box where it has 2 chambers, and it can make a choice between a new intruder, so a new animal that it can interact with, or a new toy, so a neutral object. And now if we stimulate our drug release from our magnetic liposomes, and the drug stimulates this social circuit, we can see how mice begin to prefer interacting with other mice rather than exploring these novel objects. So we can control their activity as they're behaving. And this is a strong effect. We can significantly change the preference of these mice for social chambers. But liposomes is not the only molecules, not the only objects that we can control through a local temperature increase in magnetic nanoparticles. For instance, we can surround our nanoparticles by polymers, and we can tune the chemistry of those polymers such that their hydrolysis will be controlled by a temperature increase at different rate. And if we can control polymer hydrolysis, that means we can control proton release. And if we can control proton release, that gives us control over pH. And there's many pH-sensitive ion channels in biology, and the key class being acid-sensing ion channels, also known as ASICs. And this work, led by a talented student, Jimin Park, in this work, he has developed polymerasomes that incorporated magnetic nanoparticles of iron oxide together with either polylactic glycolic acid or polysybaric anhydride. And you can see that depending on the chemistry, we can tune the decrease in pH in response to a magnetic field exposure. And you can see that polylactic glycolic acid responds very slowly, since polymer hydrolysis doesn't have very high rate or at the temperatures that magnetic particles can deliver in magnetic field. But PSA, polysybaric anhydride, has a relatively quicker kinetics, allowing us to control pH locally. And then we can apply this magnetic control of pH to a group of 100 neurons in this case that are expressing these acid-sensing ion channels. And we will see that only when our polymerasomes have the magnetic particles, and only when we apply the magnetic field, will we see calcium influx, which is a proxy for neural excitation into these neurons. But if our polymerasomes are empty, or that don't have particles, or if we don't apply magnetic field, or if our neurons are not sensitive to local change of pH, we don't see a significant increase. I bring your attention to these control experiments where we do have a little bit of response to heat, because the concentrations of particles here are actually quite substantial, unlike in the liposome work where you just need a few particles. But we have also talked about converting magnetic fields into not only into heat, but also into forces and torques. This is not a novel concept. People have been using the concept called magnetic tweezers for decades to study a function of mechanoreceptive proteins in various biological systems. However, it has been quite challenging to translate this concept of magnetic tweezers to experiments in large populations of cells, or potentially experiments in freely moving animals. And the reason for that it's actually quite challenging to modulate mechanoreceptors. It requires fractions of a piconewton forces. And it may not sound very large, but what it translates into is either the need for really large magnetic field gradients to achieve these gradient forces on magnetic particles, and those gradients typically hundreds of Tesla per meter, so this is not something that you can safely apply at large scale. And typically magnetic tweezers require bringing magnetic coil directly to the cell membrane, which is not very scalable. Alternatively, one can consider using magnetic materials with very large magnetic moments to allow you this, again, large force on those materials. But a large magnetic particle will typically mean that it's a permanent magnet. And that means it has a permanent magnetic moment. And those particles will not be stable in solution. In fact, they're going to interact with each other through magnetic dipole-dipole interactions and fall out of solution like a rock. So how do we solve this problem? This problem has been addressed by a talented former postdoc who is now a professor in Dresden, Dr. Daniela Griguritz, who has considered a new class of particles, drawing inspiration from magnetic memory, where people have been studying a magnetic state called the vortex, where all the spins of all the atomic spins within iron oxide or other magnetic materials can line up in the circular arrangement, resulting in a zero net magnetization. And we can measure this state in disc-shaped particles using electron holography, demonstrating to ourselves indeed that we see vortices. And we can see that the magnetic field around these particles is also quite negligible, which means they do not have external magnetization, and they're not going to stick to each other in solution. We can then but when we take this anisotropic magnetic nanodisk particles and we expose them to weak magnetic fields on the order of tens of millitesla, we can see a phase transition from a vortex state to an in-plane magnetization state. And when we have an in-plane magnetization state, we have a magnetic moment that is proportional to the volume of our particle. So now we can have a lever with which we can pull on cell membranes. So now we can take these particles and decorate them onto neural membranes. What you're seeing here is a scanning electron micrograph of dorsal root ganglion neurons. Those are sensory neurons from structures that are close to the spinal cord. And these sensory neurons naturally express many mechanoreceptive ion channels such as piezo1 and 2, as well as other putative mechanoreceptors. And then when we expose these sensory neural cultures coated with magnetic nanodisks to very weak, very slow magnetic fields, you can see that they begin to light up. That means they're pumping calcium into them. That means they're getting excited. And these fields are much, much easier to produce than those rapidly alternating magnetic fields that we used for magnetothermal response. Those other fields require sophisticated custom electronics that we design in our group, while these experiments you can conduct with a simple audio amplifiers, for example, one for an electric guitar and a bobbin of wire. So this is a promising technique and can be scaled up to large volumes, and we're interested in exploring it for future therapeutic or behavioral experiments. So far we have talked about converting various magnetic stimuli into physical stimuli as well as delivery of neurochemicals. But the neurochemicals become easy to deliver when they're either soluble or if they're ions. But what if we're dealing with a neurochemical that is unstable, that we cannot simply package into a liposome and then release it on demand? And one of those examples of such unstable neurochemicals is nitric oxide, which happens to be a very important neural messenger that is controls formation of synapses, controls learning, controls memory. So how do we study the function of this important messenger in context of whole organisms? Well, we can't just pump gas into the brain, so that's not an option. Another option is genetic strategies where you essentially remove ability of organisms to synthesize nitric oxide naturally, but that is very invasive and can have unintended consequences on the animal physiology, and it's also not localized to a particular region. But another strategy is, of course, to create molecular donors that can break up at the physiological temperatures and release nitric oxide. But in that case, the control over the kinetics is quite limited. So how do we solve those challenges? Well, talented student Jimin Park has thought about this problem from biological inspiration. And what he has learned that in nature, nitric oxide is actually produced from nitrite, which is the end product of nitric oxide metabolism, and it is a biological sink for nitric oxide. It's non-toxic. And the enzymes that catalyze the reduction of nitrite into nitric oxide contain an interesting key element, which is our iron sulfide cluster. And all neurons, including nitrogenases and other nitrogen fixation enzymes, contain those iron sulfide clusters. So what Jimin thought that we could do is mimic this biological catalysis using an artificial nanoscale system. And what teaming up with another trainee, Kyosuk Jin, in Cartesian theorem group, also at MIT, we synthesized iron sulfide clusters or iron sulfide clusters doped with platinum, which are known electrocatalysts. So you can see the electron microscope of those small structures on this image, and then we can confirm that indeed these iron sulfur clusters can electrically catalyze in an electrochemical cell, essentially a battery, the reaction of reduction of nitride, sodium nitride, into nitric oxide, and addition of platinum improves this electrocatalytic ability. But how do we test that, in fact, the nitric oxide synthesized in the electrocatalytic cell has biological activity? We take our electrocatalytic cell, which is about 5 centimeters or a couple of inches in diameter, and we then take the cells that we plate onto a little cover slip, and we put them next to the cathode of our electrocatalytic cell, and then we drive the synthesis of nitric oxide. We can then test this method on a test bed of neurons, which we can make sensitive to nitric oxide through expression of TRPV1 channel, which we have talked about when we talked about heating. So it happens to be sensitive to capsaicin, so the spice in peppers, to heat, but also to nitric oxide conveniently. And when nitric oxide binds the pore of TRPV1 channel, then it can transport calcium into the cells, and that will activate those neurons to fire action potentials, and we can record it here as an image. And you can see that indeed we see a response to nitric oxide generation in these cells. We can also look at the natural, non-genetically modified population of cells, how they respond to nitric oxide synthesis. For that, we take cortical neurons that contain a soluble guanylate cyclase cascade. They synthesize cyclic GMP in response to nitric oxide that can simply diffuse passively through their membranes. And what we see that indeed, that when we run electrocatalysis in the presence of sodium nitride, we see increased synthesis of the cyclic GMP in these cortical cells. However, if we don't apply any voltage, so we don't run electrocatalysis, or if we don't have our precursor, our sodium nitride, we also do not see this increased synthesis of this molecule. But okay, we can now make nitric oxide in a dish in an electrocatalytic cell. But what we really are interested in studying the role of this molecule in a biological environment. We want to do it in vivo. But our electrocatalytic cell is 5 centimeters in diameter. It's not going to fit into mouse's brain or any other brain for that matter. So we need to scale it down. And for that, we create devices that are in the form of fibers. And those fibers are made such that we can draw them in a custom process with the electrodes that will act as our electrocatalytic electrodes, as well as a microfluidic channel that will supply our precursor, sodium nitride, into the brain. These devices are quite miniature. Those fibers are a couple of 100 microns in diameter, and they contain 2 electrodes, a gold-coated tungsten and platinum-coated tungsten, and we coat our platinum, our iron sulfide clusters onto gold-coated tungsten electrodes, creating a cathode, and then the counter electrode is coated with platinum. And then we can implant these miniature devices into a deep brain region, again into ventral tegmental area of live mice. And then we can measure calcium influx into cells in regions that are downstream from that region, such as nucleus accumbens or medial prefrontal cortex. And what we find is that if we, in fact, run electrocatalysis using these fibers implanted into the brain, once we reach a threshold voltage of about 2 volts and starting around two walls a little bit more, we start seeing neural activity manifested in this increase of calcium signaling in this circuit. But without nitric oxide supply, so without our precursor sodium nitrite, or without sensitivity to these nitric oxide signaling, we do not have this kind of calcium influx. So we now have created a system that was based on iron sulfur nanocatalysts that can now be used to control a gaseous messenger nitric oxide in vivo in live animals for the first time. So let's bring it back. So what we have so far talked about is that if we really want to understand neural signaling, and we need to zoom in to the level of the nanoscale. We need to understand the signaling of individual molecules, receptors, ion channels. And because those molecules are nanoscale objects, the only way for us to really control their function is to create nanomaterials that can operate on the same spatial scales. But then we need to be able to deploy those nanoscale solutions in macro scale systems, such as behaving animals, or in future potentially human patients, to really leverage nanotechnology to both link the molecular and behavioral neuroscience, understand the role of molecules in behavior, but also to create new therapeutics, for example, therapeutics for Parkinson's disease and other neurological conditions that can benefit from minimally invasive approaches for modulating their neural activity, because nanotechnology is not like a solid device that produces damage. In fact, it can act locally, modulating only its intended targets. So at this point, I would like to thank my talented group of students and postdocs and highlighted trainees who are responsible for a lot of the data that I have presented today, as well as a number of really gifted alumni who are now starting their own groups in different institutions across the world, and funding sources that make all this work possible. And of course, thank you very much, Nature Nano. Congratulations, it's a huge accomplishment, 15 years and going strong. Thank you.
Thank you very much, Paulina. This was really a fascinating talk, and thank you for giving an overview of what you're working on. There is a question from the audience. So someone asked if you can give further details about the biocompatibility of metal oxide.
Of course. So we work with iron oxides and also with tertiary ferrites. So and iron oxide magnetite that we typically work with is quite biocompatible. So we test biocompatibility ourselves, but it also has been studied by many other groups. It actually has been in a clinical trial for glioblastoma treatment for hyperthermia in humans. And so it is a benign sort of biologically inert material. However, of course, if we want to tune properties, we can add other ions, we can add nickel, zinc, cobalt, and for those materials, we need to start worrying about their biocompatibility, which we do in our work, and we report that data, typically running various live dead assays as well as proliferation assays, and we find that particles are more biocompatible in vivo. In fact, we can rarely find immune responses to those particles, unlike large-scale devices that are sort of operate on the macro scale and cause mechanical damage to local circuits.
Right. Do you, like, following on this, do you follow the fate of the nanoparticles, or is it once it's been you see that it's biocompatible, then that's fine, or do you like actually look at what happens to them?
Well, this is definitely a work in progress, and it's an excellent question. So we have followed the fate of our nanoparticles in the context of magnetothermal stimulation, both in the brain as well as in the adrenal gland, in the work that I haven't had a chance to talk about, but it's also published. And there we found that our particles are stable, and exactly in this location where we have injected them into 3 to 6 months following the injection. And in fact, we think that's probably going to be longer, but mice ultimately are not very long-living organisms. So we will have to run those experiments in larger models with longer lifespans. It is definitely still work in progress. We, for instance, have not looked at cell internalization of the particles in vivo, so we haven't done that electron microscopy yet. For our liposome work for drug release, those are different chemistry particles, and they're coated and they're in lipid vesicles, and we have also worked with much smaller concentrations. So those particles actually do get sequestered out, and they are cleared out of the body in about I want to say within about a month, depending on the concentration. So there, but the scale of the injection is very different, and the chemistry is quite different. So for just simple magnetothermal stimulation, our surfaces of the particles are coated with polyethylene glycol, which makes them really inert. But when we are putting them inside a lipid carrier, lipids, of course, have affinity for membranes and are more likely to be internalized.
Thank you. There is another question from the audience. So the question is it possible to target particular neuronal networks, for example, dopaminergic neurons, with nanoparticles in the way as it is done to certain type of tumors?
So tumor targeting often relies on either signaling molecules that tumors present outside on their membranes, or on the fact that tumors are much more vascularized. So for neurons, things are a little bit more complicated, because neurons very often would present similar molecules on their membranes, and they also have sort of normal vascularization that doesn't make them sort of more susceptible to particle accumulation. So we need to be a little bit more clever in our targeting. This is the work that we're very interested in right now, and is the next step for our development. There you can either look for antibody-based strategies for targeting more selective moieties on neuronal membranes. Alternatively, you can of course go with genetic routes, and which works in an animal model where you can express a genetic moiety that would present itself exclusively in the cells of interest. And we're pursuing both of those routes. And of course, there is some other hybrid strategies that we can pursue, which is starting to think about magnetic localization and guidance with the combination of some chemical targeting. But it's definitely, this is a very hard question, and probably will keep us busy for another decade.
Yes. I have another question regarding the magnetic nanodisk, so the mechano control, let's say, of the transmission. So you showed that you actually applied the nanodisks on neurons. So that's, I think, the only way you have at the moment to apply that type of stimuli. Did you see any, does it affect neurons in any ways? Did you see any toxicity of the material? Is it something that, you can think of for potential translational avenues, or is this like mainly to understand how you can apply that type of stimuli to neurons at the moment?
So for magnetomechanics, we have looked at the health of neurons sort of upon functionalization with particles, as well as following application of magnetic field. And the conditions that we chose for those experiments, and we have looked at the sort of different field magnitudes and different frequencies, do not cause any, let's say, membrane rupture or other adverse effects on the cell health. However, it is of course possible to apply more field and target a bigger disk and tear the membrane apart. And that has been, in fact, done about a decade ago by, I think, a very talented group from, I believe, from Akron or one of other national labs, where I have applied particles to rupture tumors in a sort of this mechanical way. However, we specifically work with somewhat smaller particle and at much more gentle field conditions, so that we don't see those types of effects. Targeting is, again, key challenge. We always come back to targeting. The experiments that we have done were electrostatic targeting, so it's essentially non-selective. It goes after all neural membranes, and of course we'll get other cells as well. So now we are reevaluating this method again to make it selective to particular groups of neurons, and translating it in vivo, because all the experiments that I have shown you so far have been in vitro. And that's basically the next step. And so some of the targeting strategies that I have described, we are deploying them in the context of these particles.
All right, thank you. And I guess just to wrap up, I wanted to ask a very general and probably like really open-ended question: is where do you see this field going? So how do you see, you talked about potential, application for developing therapeutics for Parkinson's disease or for neurological diseases. So which one do you think is the most promising strategy, and how far do you think it is from, moving towards the translation direction?
So this is of course a very, very, very loaded question. So it's very hard for me to pick favorites here, especially in the context of incomplete knowledge. So we know a lot about magnetothermal stimulation. This is something that we have been doing for a very long time. So this is the method that that is quite robust now. And we also have extended it to be able to stimulate multiple cell populations through magnetothermal multiplexing and so forth. But it does come with certain strings attached, and the key string there is instrumentation. And that instrumentation to apply even weak but very fast alternating magnetic fields is not so trivial. So we have, of course, published all our designs, and they're openly available, but it requires a custom construction of that instrumentation, or you can buy instruments that are maybe not as capable at of different conditions. If that instrumentation becomes more widely available, then this method will become definitely something that is very much ready for deployment. And as you can see, we already have some therapeutic promise from it. And the central nervous system is of course quite a big concern from a safety standpoint, but in the peripheral nervous system or other electrogenic organs, we can already see, kind of envision the therapeutic potential, because those organs are quite difficult to access with traditional implantable technologies. So for instance, we have done work in the adrenal gland, which is a mobile and really tiny organ that you cannot put hardware into. But magnetothermal stimulation allows us to trigger natural receptors in those cells and release adrenaline, release corticosterone on demand. And you can see how we can use that to treat stress disorders. But if we were to spend additional time and develop better materials and sort of develop more sophisticated understanding in the context, for instance, magnetomechanics, their electronics are much simpler. Everything will be a lot more scalable, and potentially would make a lot of the same strategies just easier and just more sort of executable at the scale of a human body. You would not need very large amplifiers. You would not need any special electronics. However, we are at an infancy of this field, so there we still have a lot of work ahead of us in terms of developing the materials, understanding how those materials interact with the mechanoreceptors, and translating those technologies into sort of animal scale experiments, and sort of looking and looking at their safety. So that's of course kind of the frontier there. And then of course we can start thinking, sky's the limit. There's many receptors, many proteins in the body. They all respond to various signals, and magnetic field is there to oblige. We can convert essentially magnetic field into pretty much any other physical stimulus we can imagine using clever materials design. And this is another field that myself, as a material scientist at heart and non-technologist at heart, I'm very interested in, and we're going to pursue. So there is kind of the the tedious but well-established strategy. There is kind of the next immediate steps, and then there is the frontier. So those are the diverging paths. And because I'm an academic and we have difficulty focusing, we're going to pursue them all.
That's great. I'm really looking forward to see what you'll do next. And thank you very much for sharing this research with us. It was really exciting. And it's time really to close. So thank you also very much to all the speakers that participated to this webinar series. It was really great. And to all the audience for being with us. And I wish you all a nice end of the day, continuation of the day, night, depending on where you are. And thank you very much again, everyone, for attending. And thank you very much, Paulina, again, for this fantastic talk.
Happy birthday, Nature Nana.
Thank you very much. Bye, everyone. Bye-bye.